Yeah, it's up on the screen. There it is. Cool. All right, thank you. All right, everyone, we're going to get our meeting started. Hello. Good evening, everyone. Can you hear me in the back? All right. Thanks so much for being here. Welcome to our July meeting of the Western Pennsylvania Mushroom Club. I've got some quick updates on what we've been up to and what we have coming up. And then I'll introduce our speaker for tonight. There we go. All right, so you should have received your July newsletter in the mail recently. There's some really great articles in there. There's a recipe, mushroom risotto recipe that I think Mike wrote. Check it out. There's also a summary of the City Nature Challenge results. And yeah, we've had 4 walks since our last meeting. It's been dry, so we didn't find a ton of stuff on all of our walks, but we found some stuff on every single one, no matter, you know, how much rain there had been lately. First up, there was Deer Lakes Park. That was the Dorothy Fornoff walk. And we had a pretty good group. I think about 18 people showed up. And yeah, Richard led the walk, found a bunch of stuff there. On July 3rd, I led a walk at Wintergreen Gorge up in Erie. We had a nice crew there. My family came. On July 6th, Julie led a walk at Limbrook Woodlands. So a lot of folks came out to that one. That was more of like a beginner mushroom walk, I think. And yeah, looks like you had a lot of finds. And then probably our most successful walk recently was July 9th at Lind Run State Park. Really wish I could have made it down to that one. Over 70 species recorded. Yeah, there's some nice, beautiful Amanita jacksonii there. Is that right?
That was the prettiest find of the day.
Yeah.
That was in photo.
Cool. Got to make it down that way sometime. All right, we've got some upcoming walks. This was just announced this upcoming weekend, July 23rd, Crooked Creek Lake Park. There's a flyer I'll show you for that. And then our next walk after that will be August 13th at Boyce Mayview Park. And that's with Venture Outdoors. For that one, there's a $5 fee. It's through Venture Outdoors, and I think it's a fundraising event. It's part of their big day out or something like that. But yeah, keep an eye on our website and Facebook for pop-up walks. If we get more rain, it's pretty likely we'll lead more walks. So here's the flyer for that upcoming walk. That's this Saturday in Armstrong County. So hopefully we'll find some mushrooms. There's also some walks with Allegheny Land Trust, July 30th, Sycamore Island, August 12th at Bethel Green. Usually Julie opens some more tickets like right before those, like the week of those walks. So if you really wanted to go, I don't know about Sycamore Island, that's on a boat, so there's limited space. But probably for the Bethel Green walk, there'll be some last minute tickets. Just keep your eyes out. But yeah, keep an eye on AlleghenyLandTrust.org. They lead all kinds of nature walks, not just mushrooms. Yeah, so here's our meeting schedule, upcoming meetings. Some other updates that we wanted to share. If you really like having data slips, you can print your own now. You can go onto the Fundus and print your own field data slips. They are free. Just gotta pay to print them. You can buy our club merchandise on Spreadshirt now. So if you go to this website, you can customize different t-shirts, hoodies, bags, and have a nice little Western Pennsylvania Mushroom Club thing to carry around everywhere. I don't know. I guess I need a new reusable shopping bag. Also, this is very, very exciting, especially now that we're starting to see some boletes out there. The bolete filter app is now a thing. Yeah. I think it's worthy of applause. But yeah, I've been testing it out. It seems to be working well. And if you haven't tried it yet, give it a shot. It's very similar to what was already up on our website. So I don't know if you were like me, but I would be out there, you know, at Heartwood Acres, pulling up the Western Pennsylvania Mushroom Club website, going through the key in my browser, and it was a pain in the butt. And now it's so easy. So you have no excuse. Get out there, find some boletes, and identify. Our biggest event of the year is coming up in September, the Gary Winkoff Memorial Foray. We'll have our pre-foray walk on Friday in the forest. That's free. So it's during the day on Friday. I know not everyone can be off work, but you can be there. It's a really great walk. I made it for the first time last year. Now I'm committed. I'm going every year. On Saturday, we'll be at the Lodge in North Park for our full day of mushroom festivities. We're bringing in the Bassettes. Really excited to be hosting them. Alan is a distinguished emeritus professor of biology at Utica College of Syracuse University. He's a professional mycologist and has authored and co-authored more than 25 books. At the foray, Alan is going to be helping with mushroom identification, and he'll be giving a table talk at the end of the event. And Arlene has authored or co-authored 18 books, and she'll be giving a presentation on polypores, which is the topic of their most recent mushroom book that they published. Yeah, they'll both be available for meet and greet during the afternoon. And of course, we'll also have Stephen Russell joining us. He's the author of this book, but he's also the founder of the Hoosier Mushroom Society, and he's really paving the way for DIY DNA barcoding folks. He's trying to know all of the mushrooms of Indiana, and he's put in a lot of work sequencing collections, organizing forays. So he's going to come and about some of his work doing that. It'll be kind of similar to what Garrett will be talking to us about tonight, but I'm sure it'll be also very different. Yeah. Speaking of the foray, if you are interested in volunteering, you can reach out to the committee chairs using this email, winkoff-foray@wpamushroomclub.org. Awesome. Yeah. So this makes it to the live stream. Mike Henry said if you are interested in preparing a dish, it doesn't have to be a mushroom dish. You can reach him through this email. But yeah, we need a lot of people to sign up to bring food. We have a potluck dinner, and yeah, we provide a lot of stuff. We also will reimburse folks for what they spend on groceries. Is that true? To a certain degree? Ooh, bring food, get a free t-shirt. Yeah, so save your receipts, bring your receipt, we'll reimburse you for the cost of the groceries, and we'll give you a shirt. So yeah, if you like to cook, it again doesn't have to be a mushroom dish. The one thing is if you want to put wild mushrooms in it, you have to get them identified by one of our club identifiers or mycologists. So just keep that in mind. You can use store-bought mushrooms, don't have to talk to anyone. Do your thing, right? But if you want to use wild mushrooms, just come in our newsletter. There's a list of all the identifiers. You can reach out to any of us and we'll confirm like, yeah, that's an edible mushroom. Cook it, bring it to the foray. Any other questions? I am reaching out to Dale and may or may not be planning to continue said walk, trying to figure out how to manage crowds of people in the dark wandering through the forest. So we will get back to you. Oh, yeah, yeah. Sure, yeah, we will we'll post a link to the UV flashlight. There's a couple different models, but yeah, we'll post some links. Cool. Oh yeah.
My first time here, Eastwood Farms for the last.
We get permission from the city for educational purposes. Like for our meeting, we can pick mushrooms, identify them, and they have to stay in the park. So basically anything you pick, we can identify it, leave it out on the table, talk about them, and then we'll leave them here in the park. Yeah, absolutely. No, you have to leave your morels with me. Sorry. It was an okay morel season. But cool. All right, membership report. Last I checked, we were up to 857 members. So thanks so much for your support. Yeah. We've got a lot of folks here who've been members since the beginning of this club, some folks here who maybe just joined this year. And no matter how long you've been a member, your support means a lot to our club. And it helps us be able to do things like launch a DNA barcoding project and start storing specimens in the herbarium at the Carnegie Museum, and all these other cool projects that we're trying to do. So thanks a lot. A couple updates on our photo contest. The deadline is approaching, September 30th. Get your pictures in. You can have up to 5 entries. There's no size limit on your entries this year. So if you have a fancy camera, you can send your large megapixel pictures. And yeah, all the details you need to participate are up here on the slide, or you can go to our website. There's a whole page about the photo contest. New this year is the mushrooms in art category. So if you are more of a painter or sculptor or whatever craft you have, you can submit a picture of your work against other mushrooms in art things. And just a reminder that lichens are also fungi. So oh. All right. If you aren't a member of our club yet, here's what you need to know about our dues and some of the benefits. Yeah, I gotta update that one. We're not on Zoom anymore. Well, I guess we are on Zoom right now. Yeah. Oh, and we do have a Mushroom ID award to give away tonight. One person submitted a list for 50 mushrooms. Is Dan Stockman here? Ah, well, we found this picture of him holding a beautiful Hen of the Woods. So congratulations, Dan, if you're watching at home. I don't have your button. Yeah! Julie couldn't make it tonight. All right, next month, we're really excited to have Kara present on zombie fungi. Yeah, so we've got a nice Cordyceps miliaris there. Carol will be talking to us about all the different sorts of entomopathogenic fungi that you can find in our neck of the woods. Any other questions or announcements before I introduce Garrett?
Yeah.
I don't think so. I think the rules just say it's got to be a picture of a mushroom. Yeah, so if you travel, take pictures of mushrooms elsewhere, cool. We'd love to see them. All right, well then, let me introduce Mr. Garrett Taylor. So excited to have Garrett presenting tonight. Yeah, he's going to give us an update on his progress towards his goal of knowing all fungi in Eastern North America. Garrett has been a really dedicated community scientist. He's been out there photographing, documenting, doing microscopy, doing DNA barcoding, and publishing all of that to public databases. And he's one of the top identifiers and observers of fungi on iNaturalist for all of North America. So you may not know it, but he knows what he's talking about. He's a pretty cool guy, and I'm really excited to turn it over to him.
Thanks. I will do. Okay, talking to microphones. Well, this is good though. Also, I was out getting really hot, so that's why I'm all red and stuff. It's not like embarrassed yet. I'm sure that'll happen. Yeah, do we see like the folks at home, or is there any way to see that or get questions? There are no folks. Oh, there's some folks on Facebook.
It might be on the Facebook page.
All right.
So we can try.
Anybody's following along at home, and text somebody here that's here, and then they'll ask the question for you. I'm not really going to talk about so much like what we're doing out there and that's all technical stuff. And there's manuals on that, and it's all changing. Like we're switching to different, like next generation sequencing and stuff. So all that process is going to be different. So I don't want to even like talk about that. But what I want to talk about is what I found out, more or less. So and how to get into it too, and what I do. I go look a little bit more extreme than most people with the microscopy and all that. But a lot of people will just take cell phone pictures. I got a fancy DSLR camera. And I just advance like by pressing this red thing.
No, just press the space bar.
Okay. I don't ever do these programs either, so that's the title. Yeah, that doesn't work.
1 second. No.
I'll talk about these at the appropriate time too. I'm not going to start talking about it before the appropriate time. All right, that's the next slide. Those are just different reasons why I heard people got into fungus. And okay, we're going through.
Sorry, I'm going. Am I going
Now,
backwards
right?
or not? No.
Okay, you go backwards. How do you do that? That's too much. Back arrow.
All right. So I think press space bar
Okay.
and you're good.
Okay. Well, you can read all these things. That's the reasons that I've heard of. There's probably more. Feel free to interrupt me if you think of something to add, because I'm not in the right frame of mind. Being outside and getting real hot, it doesn't put you in the right frame of mind to give like real everything on the boards here. So I don't want to leave anything on the table, but also I want to go through this kind of quickly because it's kind of a long program. If I can get wordy too. So let me shut up. Why sequencing? Can I move myself? Oh. All right, I'm gonna have to move myself all around all day. It's just like Zoom.
It is.
It is Zoom. It's more accessible to the public and report. You guys can read all this stuff. to know these things, like that's the reason why we do it, right? We're gonna not, our guides are all wrong. Like people are writing guides now, I feel sorry for them. Like people are just gonna be poo-pooing their guides for the rest of the history that come out now. Without all this stuff, it's kind of hard to write a guide because everything's different. I think that the third one down is kind of the technical stuff, that names are no longer able to be applied due to biogeography. Like we have different stuff that's it than is in Europe, which everything was named, we took the names. Everybody knows that one. There's a lot of hidden diversity, so we have one name for things. There's 7 names for, or 7 different things that go under that one name. And sometimes people name the same thing 7 different times. So that happens too. We don't know these things until we get all the type sequences, and that's a whole nother issue. So there's a lot of stuff that, like, when I sequence something, I take it to GenBank. I'm sure I talked about that here. It's been a little while since I've given this talk, and I changed the slides last time. I haven't really looked at it too much lately, but this is the whole tree of life, and everything has a sequence. We are way down here. I have a little pointer actually. We are way down here in Aphistoconta. That's all the stuff we're looking at is in that as well with the humans like us. So we are all together. It's like right there. That one right there. That line. That's like almost all of life that you know, except for plants. No plants. So everything you see outside almost right there. Everything else microscopic. Just that's a lumping. That's a lumpering. And then in fungus, there's a whole bunch of stuff right here. I don't have my speaker notes too, so that's another thing that I'm not like, I'm just gonna wing it. So what we're looking at is that arrow. It's pointing to something called dicaria. That's the ascomyces and the basidiomyces. Everybody knows what a cup fungus is, right? Like the morels, and they kind of shoot their spores out. And everybody knows what a basidiomyces is, right? Like a polypore or a gill mushroom, where they passively drop their spores and the wind takes it. Well, the wind and the water takes everything eventually, but that's kind of how it happens. So those 2 branches, and then everything gets broken up into all these different, what are they, orders? Yeah, anyway. All this stuff, there's a whole bunch more stuff here, probably even much bigger, but like, I think they're talking about like evolutionary tree. You start with fungi. That's like the first thing that came out, probably, I think, with like plants out of the ocean. When the plants settled onto the land, they had like, I think they call them endomycorrhizal, not ectomycorrhizal, endophytic, or in the actual plant. It helped the plant come out onto land and survive. So, and then there's a whole carbon cycle that fungi are involved in. But so that was like the birth of everything. And then it followed up there. That's like the most evolved. So maybe this is like these early branches. These are stuff that are maybe a little more ancient lineages. So all the stuff at the top more evolved. If you look at any tree, it sort of mirrors that. Like, there was a big thing in 2019. I think I have a picture of it. No. Oh well, that's what trees are. I'm always looking at trees, and that's something that you do a lot of when you start sequencing. You get sequence back, you take it to the tree. Where does it fit? What am I looking at here? Sometimes it's way off of what you thought. But in the field, you guys can read all that. Hopefully, you guys read that home too. That's a little bigger. That's what the slides I have. Most of this stuff, you're not gonna, like, iNaturalist does for you, except for the permission, of course. You won't get permission. I guess we're working on that for all of Pennsylvania, though. Some certain state lands and stuff, we're gonna be able to collect for science too. Right now, it's a lot of the state land is like But how do you know if you're going to eat it unless you take it home and study it? Like, it's not like everybody isn't just born into the world knowing what you can't eat unless you're Czechoslovakia. Macrochemical tests. That's always cool. The taste is fit test is my favorite macrochemical test. And, you can be done on any mushroom as long as you spit it out. Habitat, noting habitat, that's something that I had a tough time with because I didn't know my trees or anything like that. So I have to learn all that stuff. It's a whole other thing. You learn like a whole ecology, everything. Pretty cool. So I recommend it. At home, this is the boring stuff, except for the microscopy. That can be kind of fun. But this is the whole process, and we'll go over that with you if you're collecting anything for pretty much if you come out to a 4A, we tell you the process that is involved, except for the drying. We're always trying to get people to key things out. That's a really good key right there. Really easy to follow. It's like the next generation of keys. It's like just like you start out at one wheel and you look through it and you find out where you go to the next step. You look at the next wheel and you find out where you go. And there's actually even microscopic characters in that too. So that's a really cool thing. Quickly and thoroughly drying, not more than 115. Some people say that's even too high. Pretty much need a dehydrator or a fan blowing on your mushroom. You cut it really thin, maybe put on a screen, maybe not put in the sun, that might be too hot. Okay? So those things are kind of important. Okay, I really want to help. What's next? What is next? Find the professionals projects. There's some opportunities for that, requires some specialization that might not have been acquired yet. Like you might not know what the thing the specialist is studying is. So you want to learn what those specialists are and what those things are, like the amanitas that Rod Tullis studies. That's kind of what got me going. I'll talk about that. Join a club. We have a lot of those people that are already involved submitting things and things like that. We have our own projects here at the Western Pennsylvania Mushroom Club. You can find a biohacker like Valerie or community science space, and they'll show you some of that equipment and training that you need to learn some of this stuff with a PCR and the more technical things. And then start keeping things that you and your crew can't put names on. This is probably the cheapest, the least immediately gratifying way to make a real impact. Now I say that's least immediately gratifying, that's gonna change. We're gonna have the next gen sequencing at our fingertips, hopefully more than like it's just a matter of time. And then we'll be able to know what we found on Saturday and Monday. The NAMA, that's kind of what got me going because I live in the middle of nowhere. I don't know if you saw the picture. I live way up in Bradford, Pennsylvania, which is on the border of PA and New York, and in like the Allegheny State Park. So that's where I grew up. And there's nobody that knows anything about mushrooms there. Knock on wood. No offense if there is. I'm really sorry about that. I just haven't met you yet. I would really like to. Let's go out sometime. That's sort of like got into books, and learning from books is tough. Picture matching, and it's not always in the book. So that wasn't really even a problem that I thought I had for a long time. But now it's become a huge problem. I'll tell you some of the reasons for that. One of the guys I met in my first NAMA foray at Clarion, Pennsylvania was this guy, Rod Tullis. He runs this site at the top and highlighted, imitacia.org. And he took some things from the foray home that I found, right? So this Amanita flavoconia was one of them, the yellow cap and yellow scales and nets. I think this is actually something else. This is Scenario penosa, but this is one of the ones I found on his site like a couple of years later. He took this back and did a little sequencing on it, and like my name's up at that site. Like that's pretty cool, right? So I thought that was really cool. That was back from 2011, 8-7. I guess that's probably where he sequenced it. I think our forward was a little earlier than that. Maybe not. It was around then. don't remember finding that. I remember finding this very vanilla stuff. That is pretty cool. Here's another mushroom which I happen to have right here to look at. So really good pictures. That's what I pride myself in. Really good picture, taking really good pictures. But this is actually even better than that. So you can all see it. Here's one right here in real life. It's got this cool snakeskin pattern on the site, which is kind of eaten away by the slug. Too early for it to show on that early one, but both of these were sequence specimens. That was one I sent to Rod, and this is one I did myself from a different part of the forest in the same area. I'm almost 100% sure that this, but I'm actually going to sequence this too, because amines are tough. I'm wrong a lot. All right, I think I don't know what that is. Okay, the results. So this is a project I started through Fundus, which used to be called mycoflora.org. In 2018, they gave me 33 sequences. I thought it was the coolest thing, and I saw the price. I was actually already had my PCR machine at home, but I saw the price was $10 a sequence, and they were giving free sequences away to start out. So like, well, I could pay $10 and do all the work myself, or I could just have them do all the work. And I wish I did it myself to start out with, because it's pretty gratifying and it's immediately gratifying, whereas the results sometimes take up to a year. I have results I've been waiting on for 2 years. I'm proud about. That's a different story. He's going to make it up to me though. 90% of the results I got back, I say 2018, but this is sort of like all the way up until now. Like, you think that with seed sequencing, thousands of mushrooms every year from Indiana, with everybody in Fundus that was sequencing, there wasn't that many of us, but there should be more of us. With Western Pennsylvania, whoever submitted everything to GenBank, 90% of the submissions returned with associated sequence data. Fewer too short to submit or contaminated. So 90% success rate. 10% I have to do again. So I still have those. I'll probably redo them at some point. 40% came back not named, so they didn't have a name in GenBank. That's actually a lot lower now. I think it's more like 50%. About 50% come back named in GenBank. About 15, more like 20% come back, and not only are they unnamed in GenBank, they don't have a sequence that matches in GenBank. There's a lot of stuff that GenBank has in there with temp codes on them, or like environmental sampling where they take soil and they put that all through. And then 8% is we're just sort of guess. Maybe they're new to science, maybe it's less than that. But so like out of every 10 mushrooms you find, you almost find one that's like new to science. At least, you know, I'm not going to say you're going to go out and do that in your first foray or anything, but eventually you'll get to the point, and then you'll probably surpass that. Who knows? But maybe we'll have them all named by then, with our stepping up with the next generation. Who knows? But it's cool. Here's a graph I made of duplicate sequences. Okay, duplicate sequences are the blue. So that's stuff I sequenced twice. So it starts out, and just every time I sequence one, occasionally I'll get one that I've sequenced twice. And there's a new sequences added to GenBank in the red. So those are sequences that are on GenBank, have no temporary codes whatsoever until I put it on there and put my own temporary code on there. And the number, that's the total number of sequences. So I've done like almost 600 sequences. This is another graph. Total new species. So maybe these are new. These are things like that don't have anything in GenBank. So as I get up to 600, you can see I'm up to 125 new ones. I think I missed something in there. Oh yeah, I just want to show you a couple cool ones. This is Humidicutus marginata, so I thought. it turns out is new to GenBank. So we're calling it Marginata New York 01 or something. Here it is again. This is a different one from a different place. I've got one in the Laurel Highlands from here, but most of these are from Allegheny State Park. This looks like Marginata var olivacea, but it came back the same as that one. And this looks totally different, but it came back as that from the Laurel Highlands. This is a Mycena species New York 01, or like it was close to Robusta. Now the interesting thing here, this is also Mycena species New York 01. And this is sort of bad. This is a cell phone picture. So a rare cell phone picture with a rare blurry edges. So I tend to not take cell phone pictures, but that's changing because we just need to push this stuff through. This is a cool one. This was found near some what I thought were Matsutake in the Allegheny State Park. You can see that it's got like this white stuff on the base. That's this stuff called we share. It's in this stuff called podzol. It's a middle layer. It's nutrient poor. They call it like an ashy layer almost that forms in the between these like the humus and whatever is on the bottom of the soil, I guess. And that stuff is attached where the mushroom grows. Same with Matsutake. So if you find something like that you think is a Matsutake, and it doesn't have that, it's probably not a Matsutake. But it probably doesn't smell like one either. So find out what it is. But this is Tricholoma, not Matsutake. I just said that because it was growing right next to it. It's in a mossy hemlock hummock. And I found this twice. It turns out it was new to science, and now it's getting a new name. I can't remember what the name is, but I don't think I could tell you anyway. I'm like sworn to secrecy or something. The mycobiome people have found it up in Quebec, and they have named it on their. It's like section genuina or something. There's a temporary code on their site. Big mushroom. This is the same thing. Looks a little bit different. This is a cool one that was in their paper. They talk about, just briefly mention, this is the closest thing in GenBank to a mushroom that they're naming. But this is unnamed. And I think it's new to science. Really pretty mushroom. Also Tricholoma. Cortinarius. Another one that mycobiome has taken and run with. They're working with a lot of the scientists. They're way ahead of us. They've been doing this a lot longer than we have, and this is one that they found in hickory groves. They don't have too many hickory. Well, yeah, they have hickory up there. What am I thinking of? They don't have a lot of oak up there, so they're hunting a lot of oak because they try to fill in the gaps. Their gaps are already filled in, except for the weird rare habitats. So they don't have as much work to do as far as general knowledge and stuff that somebody in the club probably knows it and can help them. But they don't have a lot of the stuff that we have. And this was one that they just found and put a name on. And I don't know what that name is. I'm probably going to tell you if I know. The glutinoglossum. This matched a species from Japan or something. Glutinoglossum orientale, but it didn't really. It was pretty. These things are like many other mushrooms. A very small difference in the DNA makes a new species. A lot of mushrooms, like they're not as diverse. There's a lot of these things. So this is almost one of those mushrooms. Just collect all of them and sequence them, because you'll probably come up with some new ones, even if you're doing the microscopy, especially if you're doing the microscopy. But this is really cool. You can't even like grab these things. This is all like glutinous. You kind of see it. Like, it looks like it's out of focus, but that's just like really slimy stem. Really cool mushroom. Never saw it. Thought I'd seen it before. but I think it was a wet day or something, and the geoblossom I was looking at felt slippery. But these ones really are the real deal. Oh, here's some. Can anybody tell what that is? Dung? Yeah, so somebody else said that too. It's porcupine poop. This is a weird thing I've never even heard of. It's related to Romaria, I think, like Lanteria or Cavina. Cavina. Really cool, really small little coral mushroom. You can see where all the babies are starting. But this is a porcupine latrine by some big rocks that we have up in the north. If you guys ever get up that way, really cool geological formations at the top of these mountains. It was an old bed of the ocean. They've eroded away, but they were like harder, so they crack and they kind of sit on top and they slide downhill eventually. But they're all these big rocks. The porcupines just love them. Okay, now, oops, I skipped that one. That's okay. It was the same as this one. I sequenced both of them. And I sequenced this one. And it was the same. And I sequenced this one. And it was also the same. And you guys can figure out what I did with this one. And that was also the same. And so was this one. And so was this one. I think I got, no, that might be it. Let's see. Yeah. So that's some of the things. Those are links. I think we can probably share this. Oh, you were first with the mask. I think we can probably arrange that. I mean, what's your goals in that? Because like, I think a lot of the actual sequencing stuff is gonna get shipped out, but we do need some help. So, I'm sure if there's help to be needed, then we'll put the call out. So just be looking out for something like that. But like, kind of have to make a commitment of some kind. Like, if we're gonna show you this stuff, we probably want you to do some of it. You know, and not just show you and then like go on your way. I know it's a big thing though. It's a process, so it's not for everyone, and you might not like it. Go ahead. I think if nobody knows it and nobody, or if there's some kind of debate whatsoever, keep it. Yeah. What did Kara say? Keep it. Yeah, you know what's up. Mm-hmm. Yeah, we don't really have a set thing where it's like, oh, this is where you submit. Like, if you have INAT picks or something that you want something to get sequenced, like, who do I send this to? Well, we probably should, right? So we probably have to talk about that and figure something out. Is there a DNA email? Okay.
So we had some things covered over but the DNA sequencing. So Garrett and Tara are going to be more future. But the simple way is I have some samples here, so you just have a grain of rice and I have a fungus slip which has the INAT number on there. And basically with those sort of things, you've got enough information to make sequence.
Yeah.
You've got the pictures. This specimen between 5 and 2 That's pretty much it.
Yeah, if you have good pictures and all different angles, it's going to be useful to somebody. So that's the biggest thing.
If you have a collaboration with Valerie,
Yep. Yeah, the audience here.
So you can do small numbers of tests. I'm volunteer to use a PCR part of them. But the
Yeah, because like all these mushrooms, like I remember this was one that I took home, and when I took home a bunch of them, I'm like, they're starting to get moldy. Like I wanted to eat some of those. Okay, so I ate a bunch. Like I better send some out to Rod. So I cut one. Like I think I ended up sending only a couple. Maybe that was a different one I'm thinking of. But I definitely ate some of them. Yeah. Yeah, when they get slimy, if there's bugs, I mean, it's if just because something's hard to ID doesn't mean it's sequence worthy. It has to be kind of in good shape, not a whole lot of bugs. But at least the structure has to still be there. It can't be real slimy, um, like a lot of dung and stuff. I mean, sometimes I'll make an exception, but normally, like it's really dry right now, we'll make exceptions right now. I'm sorry, what was the last part? Oh, how? I think we're pretty well. The ITS seems to be the most useful in that, and you can pretty much assign. You can say a temp code. This is a species. You can delimit species using the ITS, which is about between 600 and 900 base pairs long. So if you have an ITS for a mushroom, you know it's usually you can tell if it's different. Like there's some things that you can't. I guess Hevaloma is one of them. I'm sorry, and that's like I think that's over my head maybe. Yeah, you said contributing like different proteins and enzymes and stuff, but that's I think that's different parts of the DNA. Like when I say sequencing, I'm just talking about this junk region. Like I don't think it codes for much. It's the ITS region, and it sort of is free to mutate because of that. Like if you can't produce protein, you're probably not going to survive to the next generation. Well, if your ITS is kind of funky, that's just cool. That's just something that's cool about you if you're a mushroom. That's so.
the IPS region is really conserved parts which were involved with protein expression.
Yeah.
And so if viable species or viable offspring, the mutations in this transcribed section space can accumulate.
Cool. That's better than I can say. Yes. Mhm. This is getting a little over my head. But yeah, we just sequenced the ITS. That's all I know about it. So yeah, that's if you look at GenBank, most of the sequences that are in GenBank for the questions are ITS. So I know that I've had some LSU done on some of my finds for like a continental mycoblitz, and you won't have that more often than not. You're lucky if you find something the same gene.
The IPS is species, but some people want to be able to build better trees, and that's what they.
Yeah. Computers and all that. So I can't look at what a computer, I can't see what a computer sees. All right, so these are some cool links. You'll have to get to me to share this, so you can go back and find them if you're into that finding stuff. That's at the end of the one slide. That's kind of hard to do. Well, so that's it. If anybody else has any more questions, we're out there running job machine right now. We had a couple of things that we wanted to sequence, but we also wanted to make sure it's PCR machine, because we're going to be running this at this fall. So if you come to the 4A, you will have stuff sequenced. Yeah. So either one, hey, yeah, that's great. Let's do it. So my favorite genus is probably still the Amanita. Yeah, I do love those. But I like polypores. That it's just like it doesn't stop. So I like it all. Yeah, boletes. I started out with boletes, but I haven't found one that I'm like, no, about that. I think that's inedible. Edible. I'm sorry. I don't want to start. But yeah.
Mhm.
Oh yeah, I heard about that. Right. Yeah, I probably like that. Cool. Yeah, we just Michael Pollan's Change Your Mind on Netflix. Yeah, anything about mushrooms, watch everything. There's a whole bunch of mushrooms in the time of coronavirus. There's lots of and stuff. So if you want to hear those, and not just like the guy in the field that does it, that's where you go for that. Like DNA sequencing by Sigrid Jacob, who was here last month. She's excellent, and she has an excellent series of steps if you want to do PCR and get into that. So I know a lot of people have heard about that. That's also about what Alan Rockefeller does. He's got a lot of stuff out there too. But they're all in that thing, and that's all on the Fundus website there. And yeah, I guess that's it. Thank you. That's another file phone.
All right. Thanks for coming. I'm going to stop this live stream, and yeah, then we will.